So basically, we are here in the facility for next generation sequencing, and as one part of it, what becomes more and more important for us is to also be a facility for single cell research. So the single cell research is a new thing, it's a new thinking actually, that does not accept anymore that a complex tissue, which for us is a complex system, is initially destroyed so that we can take the information out. That is an aspect that we do not accept anymore, and we try to separate the individual cells from a complex tissue to individual reaction volumes, to then sequence this material. We would gain and are gaining now a lot of more information due to that technique. So that is called single cell genomics, basically. So that is basically it. So it's a technique that makes it possible that instead of destroying the tissue in the beginning, one gets to the individual parts of the tissue, which is in biological systems, the single cells.
So cool. So my name is Cornelius Fischer. I work here in the applied single cell genomics team. That's our whole group. We are basically grouped upon 4 different teams. One is the core team, which is next generation sequencing, and we have 2 other teams. One is based on applied single cell genomics, and the other team is group. You produce such a lot of data. You need people to at least help to analyze, to at least do the primary analysis of those data, where we have another team for. And why single cell matters, I usually explain like this, that if you want to understand the complex systems, the most intuitive, what we did traditionally for the last decades was kind of, if we would have never seen a telephone in our whole life and would try to understand it, traditionally what we did was we would make this telephone with all its materials and content. We would make it to a powder, crush it down to a powder, and then analyze the powder to get to the idea of what the function of it is. So, but of course, one could understand, you could follow that, but it's a much better approach to, and children do this very intuitively, that you would actually disassemble the telephone to its individual parts, right? You would then have the individual parts instead of a powder, and would then, the next thing you would do is you would probably sort it according to its look, according to its morphology, we would say in biology, and would therefore come closer to the understanding of what that complex system is for. If we would find out that it's a telephone, still not clear, but still we would get much more about the function of how that works, right? Same is for single cells. So we have a complex biological system, so we have a complex community of different cell types working together, making multicellular life possible, basically. And what we traditionally did, we destroyed it in the very beginning. So the first thing is we would lyse the cells to get to the material in sufficient high amounts. That is the DNA or the mRNA, the molecules that finally give rise to the proteins that make the function of our cells, finally. What we did is we destroyed it, the individual cells, and finally get out one vector of information, which is for this particular sample that used to consist of many individual cells, but we have lost the information. For this sample, I have a vector sample, and then the expression of the genes, that means is a gene on or off, and to which extent is it on or off. Okay? So any information that could still finally give me an idea about the composition of the original complex tissue is lost. I cannot build that from the data, because the data is a single string, it's a single vector. Now, in single cell biology, what we try to do is we try to preserve the cells as intact as possible. Put them in individual
rooms, individual reaction volumes. This can be little physical reaction volumes that are built from metal or plastic or whatsoever. Or it can be droplets of a fluid, water basically, in oil that make up those different reaction volumes. There we can do the conversation of the DNA to information. So shortly said, to make DNA information maybe analog to a digital readout. And by doing this, by having that separate, we then have not only one vector left for this sample, but we have actually thousands of vectors. We have thousands of vectors. For every cell, we have a data vector, and for every cell, we then can detect which gene is on or off. We can detect thousands of genes, very much depending on the cell type. Can detect thousands of genes for many thousands of cells, and that then gives us the information from the original tissue. It lets us express the distance the different cells have to each other, so all cells versus all cells, based on not only a few genes, but thousands of genes. So there we say it's the distance the cells have in data space based on their whole genome, so to say. This makes it possible to see the cell types from the individual tissue as separate clusters, because from one cell type, cells from one cell type will have a relatively similar expression to each other. They form a cluster in data space. Cells from another cell type have in itself similar expression pattern. They form a cluster in data space, but they're distinct from the other cell types. And so that is interesting to look at one tissue, and now we go further and compare different situations. Compare, for example, a tumor tissue versus a part in the human body that is not diseased, that is not a tumor. And this can be put further in saying, okay, I have a tumor, I treat it with some agents where I believe it's beneficial to treat cancer, for example, and I would then see which of these little cell populations are affected. Do they increase in number? Do they change in the expression, in the gene expression they have? Or is this an increase or a decrease? And therefore I can actually interpolate or somehow find out what would be a good treatment against cancer. Cancer is one of the most interesting points for this kind of research, and also turns out if we look basically at if you look how the instruments or this whole single cell approach is used, it's at least a big part, 30–40% is in cancer research. So it's a big question how to accomplish such complex projects. So back 10 years, 20 years ago, on the whole, the methods were not easier, but they were more focused. For example, people were studying very concentrated in different fields, for example, the protein expression of some genes, are some proteins interacting, yes or no? That's super important research. This helped us a lot to understand about biological systems. But what we have now is, we have the problem that we need to have a super multidisciplinary team to solve this, okay? So this starts even, I need people that can do microscopy. I want to see in a tissue, how does the tissue look like? I need people that are good in making this tissue to a single cell suspension. So this is an important requirement, because for most techniques in the past, that was not really necessary to have this. You would destroy this tissue, and then you have the material, and then you can work with it. Now, it's different. You need a very good quality single cell suspension with individual cells being viable and healthy to be as fast as possible processed with our single cell techniques. So okay, people that can do microscopy, people that can do the sample preparation, you need people that can work with complicated instruments, with instruments that partition the cells into individual volumes or rooms. And you need people that can actually prepare things, prepare this material outcome to a so-called library, to a material that can be used finally for our sequences. Yeah? Next stage would be you need people that can run those sequences. Yeah? And with this, you have all the lab work done, but you have still not learned anything about the biological system, about the sample you had in your head. So it was, because then comes a huge job, which is most uncommon to most biologists. It's getting better and better now, it's very amazing, but most uncommon is that the same people that are in the lab working with biological material are in the same way computer experts. So you need some people that are super focused on that, super focused on working with the huge data output that those sequencers give us to do a primary analysis, to handle the data, it's a lot of data, and to finally process the data in an appropriate way where it's always different from protocol to protocol, from data analysis type to data analysis type. So it's quite interdisciplinary actually. And there you need some people that have an overview about all those steps, and probably are not so good in individual aspect, of course, yeah. But it have the overview, and you need some people super specialized. And this is for science a complete new challenge, to group together a team that has the right skill, that has the collection of people that are motivated, that have different skills. And this is kind of a new thing in molecular biology, that you really need to team up. Biologists need to speak with informaticians, need to speak with bioinformaticians, need to speak with those people that do imaging, and yeah, even very broad things that are not so much connected with the classical biology. So you need specialized team. What we have is, so I'm rather overseeing things as a kind of team leader in the applied single cell genomics. I do a lot of project planning, experimental design. I do, so to say, a risk assessment. Do I want to spend so and so much money for this and that purpose to finally find an answer to a certain biological question? Yeah? Which technique do I choose for this? Do I have the budget for it? How long does it take? Yeah? And super importantly, do I have enough people at the very end of the process that can analyze the data? Yeah? So that is so the project evaluation and initiation. That's something I do a lot. And always in the meantime, in the meantime of the project progression, trying to find the best spots where I can push the project towards the highest quality of data, the fastest turnaround in the end. And in the end also a nice roundup of the data analysis. That's what I do. This is one part. With me alone, there would be no data sets generated, no publications generated. But you need actually people who do it, who are skilled in something, and this is super important. I have a very good colleague that she does sample preparation and also the processing of the single cells towards the point of that you have material that can be sequenced. So I have another colleague that is doing this also. She does it also more in the techniques that are more custom, that are not so streamlined and so on. And finally, I have colleagues who do the sequencing itself and then passing that data on to people that can do the secondary analysis. So mostly what happens here is we receive cells, We make libraries from that for sequencing, we sequence it, we do the primary processing, and then in most cases this data is passed on to people and they look for in the data for questions for answers to their questions basically. So that is super diverse in terms of questions they have, and it's super diverse in terms of models they have. So in biology, we have a lot of different experimental model systems that can be, it doesn't always has to be a tumor I analyze. It can be a fruit fly. It can be studying brain tumors will need some knowledge about processes in the brain. Therefore, as a model system, believe it or not, we take the fruit fly, or we take C. elegans as a worm, no? Or we take other organisms, yeah? Mouse, rat, of course, much more close to human. And in these experimental models, you can genetically manipulate mostly those animals and so on to, for example, say, I expect that a protein that I found in some previous data is protective against cancer, for example. Then what would be the best test for it? I would like to switch this protein off and see what is the outcome. Do the mouse has developed cancer earlier, yes or no, and so on. This is what is meant by genetic manipulation in this case. Yeah, you would, in a targeted way, switch on or off, or quantitatively change the expression, the production of a certain protein. Yeah? So, and then you can compare one animal that does it with another animal that is normal, for example. Yeah? You can ask over time how this develops from mice that are confronted with some treatment for 1 hour, 2 hours, for weeks, for months even. Yeah? You can ask super easy questions also without any manipulation. Is, for example, you take a very young mouse, some organ of a very young mouse, some organ of a very old mouse, get the single cell information out and ask what is actually changing between the complex composition of a tissue. And this may be any tissue. This may be the brain, this may be the heart, this may be the kidney, and so on. And that is, as a concept, this is very old. But as said, now we can really move to a very, very fine resolution regarding that. On the Barcelona meeting was this new way of analyzing tissue slides. It's not exactly single cell, but it adds a complete new aspect. It's called spatial transcriptomics, right? So it's when we have single cells, we always more or less lose the spatial context of how the cells work, right? We have to make them single. They're swimming in the liquid, in the buffer, we say, physiological liquid. They're swimming in there, so the original location, how an epithelial cell was placed towards an immune cell or whatsoever, is completely lost. So I mean, this is because we have to make them single to analyze them. And this was a long time now, for several years, about a decade even, this was the case. Yeah? So we here in this very new field move already to the next field, because we have something very, very interesting now, which is called spatial transcriptomics. So this is, I said, not single cell, but it gives back the single cell data its spatial annotation, or its spatial coordination system, so to say. Yeah? So when you would have a tissue slide, you are now able, or we're becoming able, and this is released, it's existing, the technique as such is existing already, but it's brought to a new level now by a bigger company, by one of the most successful company in this field, 10x Genomics. They have microscopy slides, so to say, but on the microscopy slides
is small dots printed basically. And those small spots contain small DNA molecules that are there in clusters. And those DNA molecules have the ability to fetch mRNAs, which is the messengers and proxies for us to estimate how protein expression is in a given part of a tissue slide, for example. So those molecules can fetch those mRNAs, and then you have the situation that you can actually make a picture, not with a microscope from the tissue slide, but you can actually make a picture that consists out of data. Instead of looking at the tissue slide and see different structures and so on based on some morphology, you then see in every micrometer the expression of thousands of genes. This is made by a trick. You have this probes there, this DNA probe that can fetch those mRNAs. But on the same time, for every of the 1,000 points that are on this slide, where the tissue was put on, for every of the 1,000 points, you have a sequence code, a sequence barcode, a sequence index, we say, that makes it possible to finally, in the final data, in between, everything was mixed together. But in the end, we can associate the data that we find and can say this data comes from this little spot. It's one of thousands of spots, but it comes from this little spot. So I know in the tissue where I have made a photograph before, a microscopic photo, I know that at the exact location in the tissue, on the micrometer exact, so to say, which genes are expressed. I can see this for thousands of genes. I can, for example, look at the tissue without knowing anything about morphology or so. Yeah? Can look at the tissue and can ask, in this region of the tissue, is there any genes expressed that have anything to do with cancer, for example? Yeah? I can take hundreds of genes that are known to be differentially expressed in cancer and can ask where are they expressed on this tissue, and then can overlay this on the image. That's really revolutionary for for us scientists, because we could do this. We did add that, but we were always restricted for one tissue slide and then for a handful of genes. Now we have for every very small part for thousands of genes this information. That's super exciting, and it will not only make a change for research. I believe that this will make a super fast change for diagnostics, and will come super fast into therapy. You will take a tissue probe from a tumor or from a healthy tissue where you ask yourself, is this cancer, yes or no? And then you will know. You will have the signature. You will not even know, is it cancer? You will immediately know which type of cancer is it. Is it a cancer that will build metastasis, and so on. So and this can be brought to a lot of different diseases. Wherever you do a biopsy or take some tissue, you can analyze those tissue within 1 day on all its genes, and will be able to combine this with the traditional medical ways of how this was screened already for decades. Right? It's super fascinating. Spatial transcriptomics.
I mean, in the end of the day, you ask yourself, of course, so why am I doing it? What makes sense in it? So what it is? So the motivation. I mean, that's the part, isn't it? So, the question is, so for me, that is, on the one hand, it's really motivation. Is that I'm fascinated by life, so fascinated by biology, and then even more fascinated. Not only describing something, but trying to basically understand how things work. Yeah? And what fascinates me the most is those indirect ways how we get down to the function of systems. Yeah? So, this indirect way of making the transition from something living and material in a way
towards the digital information that gives us help to understand why is somebody becoming sick. How can we find out early? How can we diagnose this? So this is something what is super interesting. You don't see it from the outside, but the signature is already in the system to tell something how the system will behave in the future. Will this person have cancer or other chronic diseases? How can one predict? What can one do about it is the next thing, of course. And this indirect proof, connecting those signatures to something that confronts us all in our life. Yeah. So this I found super interesting. And whenever I see something, and even very simple processes, like how in development, how brain develops, how to see this in the data and to at least be some of the first person seeing that is, I find, super interesting thing. What is the application of this? I mean, this is another thing. I mean, from the motivation, this is now relatively selfish. Yeah, so I just like to do it. It's like a game. So we have the ability. It's super fancy. We can find out. Fascinating. Yeah. So, and probably it's a lot of people, and including me, are driven by this. Yeah. So it's fascination to see something hidden information in biological life. So, and it's maybe not even primarily to cure somebody or so. So this is, but in the end, that is what really makes the impact. Where I first really got close to this, coming from labs, from biological labs, basically, where you have some samples that are connected to medical issues and so on. So this is, you cannot really digest this. But if you first time are in a hospital, see the patients being carried around that have cancer, see how they look like, look in their eyes, then you really begin to understand the impact this has, So there's some people that know they will have to live, I don't know, for a few weeks still. And of course, I have the background knowledge and see, okay, our science will make it possible that those people will have additional years to live. Or, what is most important for some people, in some time to go, when the research progresses, we will have a super good ability to tell how long they have to live, what we have to do to make this life longer in a way, or more suitable. Some very important point there is just the prognosis. To say, okay, I have a prognosis, it's super severe, but you have 2 years to live. And this is also something what pushed me now a little bit, that I found, okay, this actually is really serious stuff what we're doing. So it affects a lot of people. So it may be simple things, but we are just before a border, accessing a new level in biology, where all those techniques that we do here fancy with the robot and stuff, that this is super routine in small devices and can be used in the hospital. So this is, and therefore, so it's a huge benefit of the people, and this motivates me too. Now we will see, and I don't know, it will take some years, but we will see that in surgeries, the doctors will take some tissue parts. For example, when they do surgeries in the brain and so on, they don't know where. It is super important. If I take out a tumor from the brain and I do not want that the tumor comes back, I need to take out some material, okay? So, which is necessary for the person to live, to continue living. But of course, I do not want to take more out than necessary. So this, and to understand this now again, it's not only how a tissue looks like in the brain or so, but it could be, and this will become sooner or later, but rather sooner, I think the next years, this will come to the clinic, that the
doctor would take out a piece where he is not quite sure, is this part of the tumor or not, and will put it for direct sequencing. So we will have this CSI-like situation, where we have, take it out, sequence it, it will be super fast, it will be in 20 minutes, you have the information, yes, that's a tumor, take it out further, or no, here, there's nothing to, it's not a tumor, so leave it in. So yeah, so then this will become happening. Yeah? This will be a big part. But beside of all those very clinical and partly sad things, what fascinates me also is this science itself of aesthetic, of life itself. So what makes different cells different? Why are they forming a community, and how do they do this complex behavior of making somebody healthy or keeping somebody healthy? And if one wants to go further, that's one super interesting project that we do is going completely away now from this medical field, rather having a look at some very fundamental aesthetic and deep problems, like how is a flower developing? Yeah. How is the flower of a plant developing? Probably there's some cells that have a very high potential to divide and do different parts of the flower. And it's a super aesthetic topic, right? So it's, and it's not solved yet. So with the single cell information, we are just onto it to solve how a flower develops, how a seedling develops, and so on. And this fascinates me also, to see something that is a part in my life, that I like plants, I like how they grow, I like how they develop, and I find it super aesthetic and super fascinating that they build up beautiful leaves and beautiful flowers and so on, and it's at the very deep core of what is life. We were always so much confronted as human beings with nature, and I never walk through a forest just saying, okay, some trees, leave me alone. In most cases I don't, but I'm always super fascinated by the aesthetics. The aesthetics is something also that drives me. So I come back from the forest completely recovered and say, okay, this was a nice trip. This was a nice trip through the aesthetics of nature.